Okay, so now we're going to start chapter 3. The first section of chapter 3 will be cells and organelles, and then our second section we'll get into at another lecture. So, cell theory. Cell theory is something that I definitely want you guys to know. It is very important. You should have seen this before in biology in high school, and maybe even in biology in college. So, there are 3 components of the cell theory. The first one is cells are the building blocks of all plants and animals. All new cells come from the division of pre-existing cells, and cells are the smallest living units that carry out all vital physiological functions. So, cell theory is without cells, we would not have life. Every living organism, plants, animals, humans, they all have to have cells. And to create that living organism, those cells have to have the ability to divide and create new cells to create that organism and to keep that organism alive. And then lastly, they are the smallest living units that carry out all vital physiological functions. Cells are building blocks of our body. So they are in itself their own little ecosystem, and they perform vital functions by themselves to keep the cell level alive, to keep the tissue level alive, and then the tissue level to keep the organ level alive, and then the organ level to keep the organism alive as well. So cells maintain homeostasis individually, but work in coordination to maintain homeostasis at higher levels of organization. So cells differentiate and specialize. This is governed by gene expression. All human cells come from a single fertilized ovum. So there at the top of that picture, there's that fertilized ovum. You see the sperm cell going into that ovum to fertilize that egg. So the fertilized ovum or zygote contains the genetic potential to become any cell in the body. So at that immediate time of conception, that is a single cell that has the ability to divide and create all of our different types of cells in our body. The cell differentiation, when it first occurs, new cells do not grow. They subdivide into smaller parcels. These parcels, which are called daughter cells, differ from one another. So the daughter cells can go into one of 4 cellular divisions in the human body. So we have our epithelial tissue, our connective tissue, our muscle tissue, and our neural tissue. That cellular differentiation, the daughter cells begin to develop specialized structural and functional characteristics of each of these 4 different types of tissues. So we have 2 groups of organelles, the intracellular structures with a specific function. Okay, so this is a very busy slide. There's a lot of information on this slide. So this triangle type box here, this represents our cell and what is all included in that cell. So we have the large unit, the cell, and then around the cell we have the plasma membrane, which is also called the cell membrane. This separates the cell contents, or the cytoplasm, from the extracellular fluid. So the cytoplasma, we'll just go down to that one next. The cytoplasma is the material located between the plasma membrane and the membrane surrounding the nucleus. This is a colloidal type syrup-like consistency. So it's kind of, if you take like maple syrup, that kind of sticky consistency. It's watery, but it's not quite watery. This contains an abundance of proteins. So the extracellular fluid, which is also referred to as the interstitial fluid, is a watery medium surrounding the cell. So the extracellular fluid is more water-like. It's not as sticky as that cytoplasm inside. The cytoplasm can be subdivided into 2 different things, the cytosol or the organelles. The cytosol is a fluid component of the cytoplasma, which is the intracellular fluid, And the organelles are quote unquote little organs. These are intracellular structures suspended within the cytosol with specific functions. So the organelles, we have two different groups of organelles. We have non-membranous organelles and membranous organelles. The non-membranous organelles, not all completely enclosed by the membrane. These components are in direct contact with the cytosol. They include the cytoskeleton, the microvilli, the centrosols, the cilia, flagella, and the ribosomes. And then we have our membranous organelles, which are isolated from the cytosol by a phospholipid membrane. So remember that micelle and all those phospholipids with the hydrophilic head and the hydrophobic tail. These guys are surrounding these organelles. This includes the mitochondria, the nucleus, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and the peroxisomes. So this is a more realistic cartoon version of what a cell would look like. So we have our peroxisomes and our lysosomes there, which are those little tiny, the lysosome is a green ball, and that peroxisome is a kind of a purple kind of marble ball looking. We have our microvilli. At the top there, that are like little hairs. We have the Golgi apparatus down below, which looks like a folding of a ribbon on top of itself. Of course, we have our nucleus there in the middle. We have our endoplasmic reticulum kind of surrounding that nucleus. We have both smooth and rough endoplasmic reticulum. We have those ribosomes, which are attached to that rough endoplasmic reticulum. We have our mitochondria down there that kind of looks like a little bit of a bean. And then we have our cytoskeleton, which is just underneath that plasma membrane, which that cytoskeleton gives us that structure support of our cell. So we have our basic cell structure, which we talked about on the previous slide with that blown up image of that picture of the cell on the right hand side. The cell is surrounded by plasma membrane or a cell membrane. This is selectively permeable. This controls the entry of ions and nutrients and elimination of waste and release of secretions. So the plasma membrane or the cell membrane is the gatekeeper to the inside of the cell. It's going to determine what things come in and what things come out, and that will help determine different reactions and different jobs that cell is going to do. The cell is filled with the cytoplasma, which includes the cytosol and the organelles. The cytosol is the liquid part, which is mostly water and it dissolves salts and sugars. The organelles are those structures that do very specific things to keep the cell alive and contribute to its function. Like before, like we talked about, it's the mitochondria that produces that ATP, that nucleus that holds that DNA and produces that RNA for that cell division, the endoplasmic reticulum, the ribosomes, which take that RNA and create those duplications of that DNA. We have the Golgi apparatus and the microvilli. We have the lysosomes and peroxisomes. The lysosome and peroxisomes are things that will encapsulate waste and get rid of that waste. So there's so many different things that these organelles do to maintain that homeostasis of that cell. The nucleus is the largest organelle in the cell. It's filled with the nucleoplasma, which is surrounded by the nuclear envelope, which has a double membrane because we want to protect that DNA, with nuclear pores. This controls the metabolism of the cell, stores genetic information, and controls protein synthesis. So the red blood cells, this is just a fun fact, red blood cells do not have a nucleus, but skeletal muscles have many nucleus. So once again, depending on that function of that particular cell is going to determine what type of organelles it has in there, what those organelles do, and how it operates. So the nucleolus is also located inside the nucleus. It is a site of ribosome production. It's made in two parts. It has an alpha and a beta, and to fit through that nuclear pore. So the nuclear envelope is a double membrane which surrounds that nucleus. The perinuclear space separates the two layers of the nuclear envelope. The nuclear pores, 10% of the surface of the nucleus, and this has passageways to allow communication between the nucleus and the cytosol. The nucleoplasma is a fluid gel-like substance. It has a nuclear matrix, which is a network of fine filaments. It provides support, contains ions, enzymes, RNA and DNA, nucleotides, and a small amount of RNA and DNA. And then we have our nuclei, which is singular for the nucleolus, is a transit nuclear organelle. It's made up of RNA, enzymes, and histidines, which are proteins. They synthesize the ribosomal RNA, and it's predominant in cells that make large amounts of proteins, for example, our liver, our nerves, and our muscle cells. So we have these vesicles that are little membrane-bound sacs that bud off the endoplasmic reticulum, or Golgi, or invaginate from the cell membrane. They are important for transport around the cell. In your book, it mentions them on page 103. So some examples of the vesicles are the lysosomes and the peroxisomes. The lysosomes are specialized vesicles that contain digestive enzymes that break down waste materials, cellular debris, and foreign invaders. And those peroxisomes are vesicles that contain enzymes to break down toxic substances like hydrogen peroxide within the cell. So these vesicles are important for the intracellular transportation. So they move materials around the cytoplasma. They also are responsible for exocytosis, which is fusing with the plasma membrane to release waste materials outside the cell. Endocytosis, so budding inwards from the plasma membrane to engulf materials from the outside, so grabbing nutrients from the outside that the cell may need. And also storage and degradation, which is holding those enzymes or materials safely from the rest of the cell. So the vesicles are really important for maintaining homeostasis in the cell as well. So the garbage crew, we talked about the peroxisomes and the lysosomes. Those peroxisomes are membranous organelles, which are phospholipid membranes, which are a vesicle. They're famous for breaking down hydrogen peroxide with the catalyst, but they also break down fatty acids and participate in synthesis of cholesterol, bile, and myelin. The lysosomes are specialized vesicles that provide an isolated environment for potentially dangerous chemical reactions. They are produced by the Golgi apparatus. They're membranous organelle. And the phospholipid membrane is it's also a vesicle. It contains digestive enzymes to break down the organic compounds, old organelles, pathogens that are mostly for recycling. An example of something that their storage system doesn't work very well, the lysosomes don't work very well, is Tay-Sachs disease, which is an inherited recessive lysosomal storage disease. So Tay-Sachs disease, it's rare and it is often fatal. It prevents the breakdown of fatty substances that lead to destructive progressive buildup in the nerve cells. It causes rapid motor and mental decline, seizures, and blindness, and usually causing death in young children. Rarely they live to kind of their teen years, and early adulthood is very rare. Currently no cure exists, and the treatment mainly focuses on supportive care to manage these symptoms. Okay, so this is the three basic functions of the lysosome. So down there at the bottom we have our Golgi apparatus in purple, and we are releasing those lysosomes there. The lysosome initially contains the inactive enzymes. Then the lysosome enzymes are activated by fusion with another vesicle or organelle. Then, as digestion occurs, the nutrients are reabsorbed for recycling, and then it is expelled by exocytosis outside the cell, the waste product that the cell no longer needs. The waste products and debris are ejected from the cell when the vesicle fuses with the plasma membrane through the process of excitosis. Then we have the extracellular solid or fluid. So we're going to grab, that vesicle is going to grab something from the outside of the cell that it needs as nutrient-wise. The vesicle containing fluid or solids may form at the surface of the cell. Once again, that lysosome enzymes are activated by fusion with another vesicle or organelle. And then as the materials or pathogens are broken down by the lysosome enzyme, its released nutrients are absorbed. So fun fact, if the lysosomal membrane breaks down the enzymes, it can destroy the cell, a process known as autolysis. So if the enzyme or the lysosome feels that cell, that little organelle is no longer working like it's supposed to, it can kill that cell by denaturalization, or that organelle because it's no longer working or functioning and maintaining homeostasis like it's supposed to. Apoptosis is similar, but the cell does it on purpose. This will come again when discussing mitosis and cancer, so we're going to get into that apoptosis a little bit later on. So we have our ribosomes, which are non-membranous. They are made up of RNA and proteins. The function of ribosomes are protein synthesis. The amino acids are assembled into polypeptides. The more proteins a cell synthesizes, the more the ribosomes it has. Liver cells have many and fat cells have few. There are two subunits that come together to form the ribosome in the cytoplasm. Those free ribosomes, they float around in the cytoplasm. The proteins made at these enter at the cytosol. The fixed ribosome attached to the endoplasmic reticulum make it rough, and these proteins will likely be exported out of the cell. So this is a little picture of that ribosomal unit. Before the protein synthesis can begin, the large and small ribosomal subunits must join together. The endoplasmic reticulum, this is a network of intracellular membranes continuous with a nuclear envelope. They have both rough and smooth endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes attached to the outside to make it rough. New proteins are chemically modified and folded here. They are packaged and shipped to the Golgi for release into the extracellular fluid. The tubular structure is continuous with the nuclear envelope, and the chambers are called cystine. The smooth endoplasmic reticulum has no ribosomes. It is responsible for lipid synthesis, including the phospholipids, cholesterol, sex hormones, and glycerides. It synthesizes the phospholipids and cholesterol needed for maintenance and growth of plasma membrane, endoplasmic reticulum, the nuclear envelope, and the Golgi apparatus. It synthesizes and stores glycerides as triglycerides in the liver for fat cells. And it synthesizes and stores glycogen in the skeletal muscles and the liver cells. So this is just a cartoon depiction of the difference between the smooth endoplasmic reticulum and the rough endoplasmic reticulum and how they differ in appearance. So you see that smooth endoplasmic reticulum, it almost looks like coral. It has got those tubular cystine in there. It's very smooth. There's no ridges or anything like that, whereas the rough endoplasmic reticulum kind of looks like folded ribbons on itself, but with all those ribosomes on the outside, which make it really rough and bumpy. Then we have our Golgi apparatus, which is also known as the body. It is looks like a stack of pita breads. Stacks of flattened membranes, there are roughly 5 or 6 of them, are called cisternines, and those names are also found in the endoplasmic reticulum. They store, alter, and package and ship products such as proteins, carbohydrates, and lipids. So that's that. We send that from the rough endoplasmic reticulum. We send those ribosomes up there with all that information. The Golgi apparatus packs it up and stores it up and sends it out to the right part of the body that it's supposed to go to. The plasma membrane is renewal, and it can be modified as well based upon what it needs to do. It modifies and packages secretions via exocytosis, which is released outside the cell. And it also releases the lysosomes as well to encapsulate that waste product and send things outside the cell as well. So the cis and trans are different sides of the Golgi apparatus. So they are the receiving side and the transport side of the Golgi. So the cis side is the receiving side and the trans side is the transport side. So we'll start there at the bottom at number 1. Proteins and the glycoproteins are delivered to the Golgi apparatus by the transport vesicles. They arrive at the cystine, which is the cis face, which is the receiving side. Then number 2, the transport vesicles combine to form the cysternine on the cis face, the inside of the Golgi apparatus. Those enzymes modify proteins and glycoproteins. Number 3, modification and packaging, migration of the cysternine and to the trans face. Then number 4, small transport vesicles return resident Golgi proteins to the cysternine of the cis face for reuse. And number 5, the product arrives at the trans face, which is the shipping size, and getting ready for exocytosis outside the cell. So now we have our mitochondria, which is our powerhouse of our cell. This is where we get our APT production. It is a double membrane. The outer membrane surrounds the organelle. The inside membrane is folded to increase the surface area for protein enzymes. These folds are called crystine. They contain their own DNA, which is mtDNA, which is mitochondrial DNA. This, like I said, mitochondria is the sole powerhouse for our ATP production. It is efficient APT production if oxygen is present, because oxygen is the final electron acceptor and permits oxidative phosphorylation. Okay, so just briefly, we're going to talk about aerobic respiration. We're going to revisit this in our muscle unit. So we're just going to touch base on it right now just to be familiar with it. There are 3 different stages of aerobic respiration. We have glycolysis, the citric acid cycle, and our electron transport chain and ATP synthesis action. So glycolysis is splitting the glucose in half in the cytoplasma. The citric acid cycle takes half of those sugars and breaks them down more into the mitochondria. And then our electron transport chain and our ATP synthesis, it uses the electrons from glycolysis and the citric acid cycle and sends them down a chain toward the oxygen. This has high electronegativity, which we did cover in chapter 2. The energy is then used to add phosphate to ADP, making ATP. So in the grand scheme of things, you're going to get a little bit of energy from that glycolysis, but you're going to use it up pretty quickly. The citric acid cycle, you're going to get a little bit more, and then that electron transport chain and ATP synthesis, you're going to get your most bang for your buck, your most energy production, and most sustained energy production. All right, so this is just a diagram, cartoon depiction of that process we just talked about. So just review this picture, be familiar with it, and understand roughly how that aerobic respiration works. All right, so now we have our cytoskeleton. The cytoskeleton is the strength and flexibility of that cell. It is basically our skeleton of our cell. It has several structures which are included in it. We have our microfilaments, we have our intermediate filaments, our thick filaments, our microtubules, centrioles, cilii, and flagella. So those microfilaments are present in most cells. They are organized in the skeletal and cardiac muscle cells. They are in bundles beneath the plasma membrane and throughout the cytoplasma, and their main function is to provide strength, alter the cell shape, and bind the cell cytoskeleton to the plasma membrane. They also can tie the cells together and are involved in muscle contraction. The intermediate filaments are present in most cells as well, and there are at least 5 different types that we are aware of. Their location is in the cytoplasma, and their main function is to provide strength and move materials throughout the cytoplasma. The thick filaments are found in the skeletal and cardiac muscle cells. They're also in the cytoplasma, and they interact with actin microfilaments to produce muscle contraction. And we're going to talk about those thin and thick filaments a little bit later on in that muscle unit as well. Our microtubules are present in most cells. They are in the cytoplasma and also radiating away from the centromere. They provide strength and move organelles. Our centrioles are 9 groups of microtubule triplets that form a short cylinder. They are in the centromere near the nucleus, and they're organized into microtubules in the spindle to move chromosomes during cell division. The cilia are also 9 groups of microtubule doublets that form a cylinder. They are motile cilia that have a central pair. They're extensions of the plasma membrane, and they have multiple moticilli that propel fluids or solids across a cell surface. They are the primary cilium of a cell detects the environmental stimuli. So they're like the little fingers on the outside of that cell membrane. And the flagellae are also 9 groups of microtubules, doublets that form a cylinder around a central pair. They're also extension of that plasma membrane, and the flagellum propels sperm. So that's the difference between the cilia and the flagella. The flagella is found on that sperm to help it swim to where it needs to go to fertilize that ovum. So protein synthesis, let's explore how a bunch of these organelles will work together to create protein synthesis. Proteins are coded for 4 genes in your DNA located by the nucleus, but can only be made in the cytoplasma. The nucleus, the ribosomes, which is made in the nucleolus, the endoplasmic reticulum, the Golgi, and the vesicles will all play important roles in protein synthesis. There's 2 major steps in protein synthesis. It's going to be transcription and translation. Transcription is making a messenger RNA copy of DNA in the nucleus. DNA should never leave the nucleus because it is a safe and protected environment for your DNA, and cells depend on their DNA to survive. That's why the nucleus has a double membrane wall to protect that DNA and make sure that DNA never gets out, because if our DNA gets out and it gets destroyed, we won't be able to replicate that DNA anymore. When a cell needs a particular protein, maybe an enzyme, it will make a messenger RNA copy of the gene in the DNA inside the nucleus. This mRNA is a set of directions for making a protein, which amino acids and in what order they're supposed to be. The messenger RNA is able to exit the nucleus through the nuclear pores, and this is special because most molecules do not have the secret passwords to enter or exit the nucleus. So only that messenger RNA is allowed to enter and exit that nucleus. So inside the nucleus, we have transcription. This results in messenger RNA. The DNA is uncoiled and unzipped through those hydrogen bonds. Remember, the hydrogen bonds are relatively weak, and so they're easy to be uncoiled and unzipped and then rebonded back together again. A complementary copy of that messenger RNA is made up of DNA, the DNA template strand by RNA polymerase, and this is a pre-messenger RNA until a 5-cap and poly A tail are added. It's now officially a messenger RNA that can exit the nucleus. So we have translation, which happens in the cytoplasma. This results in the polypeptide protein formation. So the messenger RNA attaches to the ribosome RNA. The ribosome will assemble the correct amino acid base on the codons and in the messenger mRNA to build the correct protein. The transfer RNA delivers the amino acid to the ribosome, and the anticodons are complementary to the messenger RNA codons. So the right amino acid is placed in the correct order. If the ribosome is attached to the rough endoplasmic reticulum, then the polypeptide will enter the rough endoplasmic reticulum for folding and possible chemical modification in the smooth endoplasmic reticulum before being shipped via vesicle to the Golgi and beyond. There's 3 different phases of this translation: initiation, elongation, and termination. So to the right-hand side shows an example of how that 3 phases are created with those codons and those anticodons, and then that covalent or the peptide bonding occurs. So in your Pearson's My Lab, there's lots of amazing videos, helpful study guides, but there is a really good video on this translation process and how they bind and create that transfer RNA. So initiation, the translation begins when the messenger RNA strand binds to a small ribosomal unit. The initiator transfer RNA carrying the specific amino acid binds to the start codon on the messenger RNA strand. The small and large ribosomal subunits join together and enclose the messenger RNA and the transfer RNA. Then elongation occurs, which the amino acids are added one by one to the growing polypeptide chain. Then we have termination, which is the protein releasing factor recognizes the stop codon. The ribosomal enzymes then break the bond between the polypeptide and the transfer RNA, releasing the polypeptide. Other ribosomal enzymes separate the ribosomal subunits and free the intact strand of the messenger RNA, making it available to create more copies. All right, so this is the end of our first section of chapter 3. We will be doing the second section, which is cell transport, for our next lecture.